Good afternoon, everyone. So over past hour, we've been through this journey with insertions, duplications by Luke, insertions by George Fox, and the common core by Jessica. And those patterns have been quite persistent over 4 billion of years, and we've thought everything is sort of holds in the same way. And in the next 15 or so minutes, I want to invite you to a journey into a world when all this is no longer true. And this wonderland is named Mitochondria. So I'm going to be talking about structures of mitochondrial ribosomes, and this work was done in collaboration with Alexei Amunds and Alan Brown from SkyLife Lab and Harvard Medical School and Stockholm University, and all this research was supported by Swedish Research Council and NASA Center for the Origins of Life, so I thank for funding. And as I said, so this emergence of mitochondria was dated about 2 billion of years ago, when an alpha proteobacteria basically got incorporated, invaginated into a proto-eukaryotic cell. And now it's been a hypothesis proposed by Lynn Margulis about 50 years ago, and this is now almost unanimously accepted by the scientific community, the fact that the mitochondria has evolved from an alpha proteobacteria. So, and what happened upon this process, there were many complicated different phenomena related to mutual gene transfer from mitochondria into a nucleus, as well as protein exports from cytosol into mitochondria. So, as a result of all these processes, mitochondria eventually lost its independence, and it just now became an organelle. And nevertheless, the genomic analysis performed in 90s by Michael Gray had indicated that there are a number of genomes that are always retained within mitochondria. Among them, all mitochondria essentially retain its own translational machinery. So ribosome is still in there, and there are certain critical components of oxidative phosphorylation system, such as cytochrome B, cytochrome C subunits 1 and 3. They still exist in every mitochondrial genome, so mitochondrial ribosomes are required in order to, and mitochondria from other species contain some additional variations of those genomes. So mitochondrial ribosomes are still required to do the translation within mitochondria. So, and we have this quite unusual organism. It's a eukaryotic organism called Reclinomonas americana. And it appears that it contains a genome which is a most bacterial-like genome among all known eukaryotic mitochondrial species. So this genome contains around 70 base pairs, 70 kilo base pairs, and it encodes for nearly 67 proteins. So minimal number of proteins encoded by mitochondrial genome is 4 or 5. This genome encodes 67 proteins. And it's clearly by this genomic analysis, Michael Gray had demonstrated back in the 90s that this mitochondria is closely related to an alpha proteobacteria. So, and there were a number of genomic analyses done through a past couple of decades until very recently, And this work was motivated and regained its attention in the past few years when crystal structures of mitochondrial ribosomes became available from laboratories of Nenad Ban and Venkin Ramakrishnan. So, and through the rest of my talk, I'm going to be talking about evolutionary processes that occurred within those mitochondrial mito-ribosomal structures. And apparently, they are quite different. So we have fungal mitoribosome, we have mammalian mitoribosome, and we have a parasitic mitoribosome. And they all undergo substantial changes. They accrete additional components. Most of them are proteins, such that this structure came out last year, and it's very surprising because what happened is mitoribosome nearly doubled its size compared to a bacterial ribosome, and most of it's just the proteins. It's not a ribosomal RNA. So, and let me sort of start with and maybe touch back on a question of Mike Trevisano when he asked about the common core of bacteria. It appears that there are some variations and some deletions within bacterial common core, and this is illustrated by this diagram, secondary structure. So even some bacteria do not have certain components, and mostly those are alpha proteobacteria. So for example, those regions marked in black here, they do not exist or deleted or shortened even in alpha proteobacteria. So there is a remarkable resemblance between what one can see with ribosome of alpha proteobacteria and the ribosome of Reclinomonas americana, which is mitoribosome. We have the same deletions, we have the same sequence conservation essentially. All those diagrams are mapped onto a secondary structure of E. coli. So, and that was just the common core. But now if we go and try to explore the entire array of available mito-ribosomal structures, there is a surprise that comes, and essentially common core disappears. So everything what we've talked about for 4 billion of years reduces down to an area which is highlighted in gray right here, and the common core reduces down to just to a region that which contains the PTC, a short region of an exit tunnel, and the platform of a large subunit which interacts with a small subunit. And there are a couple of other functional regions such as L11 binding domain and the sarsen-ricin loop. So. And what we did, we reconstructed phylogeny based on structures of ribosomal RNA, and I'm not going to go into details, but just to say that we will use this phylogeny in order to time the events of what happens with mitochondrial ribosomes. And when we did this and used this phylogeny and tried to map all different proteins among different species within different lineages, so mitochondria retains a number of bacterial proteins. So this mitochondria of Reclinomonas americana is the most bacterial-like mitochondria. It doesn't gain any additional proteins. There are little or no structural modification to that. All the other ribosomes, mito-ribosomes, start accreting additional proteins. And those 7 right here, ML41, 43, 45, 46, 49, 53, and 54, appear to be the oldest or nearly universal mitochondrial proteins. So when I mapped those proteins onto 3-dimensional structures, it appears that those proteins, oldest mito-ribosomal proteins, they are clustered essentially into 2 regions. One is near the exit tunnel, the second group is near the central protuberance. So let's start talking about the exit tunnel. So we actually, I mentioned earlier here that the alpha proteobacteria ribosome and ribosome of Reclinomonas americana contains some pre-deleted regions, which are marked in black here. So, and this specific region here, it's helix 16, helix 18 right here, and it's shown based on the structure of E. coli right here. Of course, there is no deletions, but if you look what happened to early proto-mitoribosome from our reconstructions, it appears that there are many early mitochondrial proteins are clustered around this region. So we hypothesize that those proteins are responsible for stabilization of this region. And then in some later mitoribosomes, so if you go to Apistoconta clades, fungi or mammalian ribosomes, this helix completely disappears. So that suggests that existence, secretion of those proteins just does a good job by itself. RNA is no longer needed. So basically, we're just talking about a complete replacement of this region. So now let's talk about the exit tunnel. The exit tunnel also gets remodeled, and it gets remodeled in two separate ways, especially when I'm talking about more advanced mitochondrial species. I imply fungi and Apistoconta, fungi and mammalian ribosomes. This is what analysis was done. So mammalian ribosomes undergo into a mode of deletion, so RNA gets shorter, shorter, and shorter, and apparently we are getting more additional proteins. In fungi ribosome, the RNA doesn't get shorter, but additional proteins get and stabilize this such that RNA itself undergoes refolding, and it causes the change in the exit tunnel path. So there is a new exit tunnel path in yeast mitochondrial ribosome, and it's diverted from its original path, vertical path, which exists in all other ribosomes, including mitochondrial ribosomes, bacterial ribosomes, etc. So, and good. So then similar events occur within another region called Central Protuberance. So we have accretion of those additional oldest proteins, ML46 and ML40. And even if George likes to talk about 5S RNA, it's being lost upon accretion of those proteins, probably because of their steric issues. And this coincides with the deletion of protein BL25. But then mitochondria gets protein ML38 accreted into the basement of this central protuberance region. And this is the reconstruction which corresponds to nearly LOCA, last Apistocontan common ancestor of mitochondrial ribosome. And from this point, the events took again two different approaches. In mammalian ribosomes, the deletion continues on, such that there is a deterioration of rRNA, and mitochondria had to do a trick. So it lacks its own rRNA, and it has to steal a tRNA molecule and just incorporate it into a central protuberance. Whereas in yeast mitochondria, there are some additional accretions, expansions that associate with additional mitoribosomal proteins and stabilize this region. So again, there are two different regimes in which those two mitochondrial ribosomes can evolve. So in order to conclude, we've shown that genetic erosion has considerable effects on mitoribosomal structure and function. Loss of mitochondrial RNA destabilizes mitoribosome, and the structural fragility of metastable intermediates makes them more receptive to accommodating new elements. Patching occurs in an onion-like fashion, where new elements are accreted onto pre-existing core. And these structural patches are the addition of species-specific pre-existing non-ribosomal macromolecules. So all those new proteins they were available. So now, if I have a minute, why all this is related to the origins of life? We are talking about mitochondria here, and I'm going back to the idea of the common core. So the nature has managed to retain the common core over 4 billion of years. The nature managed to contain the common core at the moment of Luca, when archaea split from bacteria, and yet it just completely gave up on maintaining the common core when alpha proteobacteria got incorporated in a eukaryotic cell, and it just underwent through all this remodeling. So what it suggests that there are very different types of evolutionary pressures that got applied onto this system of mitochondrial ribosomes, and it just simply recorded all differences in evolutionary pressure. And to me, the critical difference is the difference in complexity, the complexity of eukaryotic cell compared to complexity of bacteria. When there is such a difference, and it occurred over a short time, it promoted such a severe changes in the structure of ribosome, which suggests that when we got all different environmental changes, changes in metabolism, changes in cell composition upon split of Luca, all of them were not critical to the structure of the ribosome. So it survived and maintained its own common core. Okay, thank you.
Thank you, Anton. Two very quick questions.
Yeah. I have a question and a comment. Maybe the formulation of the question it would be to ask truly why the ribosomal RNA in eukaryotes and in bacteria is so conserved, in a sense, because rather than looking at what's happening in the mitoribosome, in fact, I would say that it's really illustrating that the ribosomal RNA, as you said, indeed, is under multiple other pressure, but one that is becoming very apparent when you're looking at bacteria and eukaryotes is that it's completely remodeled. The process of folding is completely controlled. There's a huge amount of modification that takes place in a sequential fashion. And in fact, the ribosome sequence is conserved also because it has to switch.
Mm-hmm. Well, so I guess the short answer to your question would be, when you need a car, do you really care whether it's a Honda or or Toyota? You don't, but they're nearly identical. They do the same functions, and all the differences are just tiny details, right?
Guys, all right, I'm sorry, we're going to have to interrupt this conversation. We have another session coming in. Thank you, Anton.